So the coronaviruses, this is an image from the beginning of the pandemic. So right at the beginning, people were focused on the 2019 novel coronavirus, and they focused on this epicenter in Wuhan, where the comparison was to previously existing viruses. So the Middle East respiratory syndrome, the severe acute respiratory syndrome, and then they were looking at symptoms and trying to understand how is this novel virus different from these other viruses that we already know and we have studied, because coronaviruses have been studied extensively for long periods of time. There are coronaviruses that infect pigs and cats and bats and all kinds of animals. They have had mild to severe outbreaks like MERS and SARS, but really they have not spread that much. And so what was new and why was it important that we understand the genomic composition of this new virus? Well, the first thing that they did was they did metagenomic sequencing. And metagenomic sequencing allows us to take all of the genomic material in a sample and then try to understand what is the actual full sequence of a particular element. So meta means a collection or a combination of many different examples. And here we're trying to understand what do these examples actually tell us about that one consensus genome that we can call the assembled genome, the full genome of this particular virus. And when they did that, they then started looking at how do we take this de novo assembled genome and start comparing it to existing genomes. So how does SARS-2 or this novel coronavirus compare to the existing coronavirus that we already know and we understand? And then we can go into biological interpretation, thinking about how can it be different? How can it function differently? How can it cause infection in a different way? So one of the critical things to understand about this is that this particular type of data that they were using, high throughput or next generation sequencing data, is actually a series of very short reads ranging anywhere between 150 and 300 base pairs in length. And they had to align it to some other reference to understand the differences. So what kinds of differences could there be? So here we have the reference. Here we have what's called coverage, or all of the reads that we have. And then we can start looking for variation. So one type of a variation is a substitution, right? So we identify here 1,213, that's the position on the genome. And we have 5 reads that all agree that this is different from what we have in the original sequence. And that's called a substitution. Another type of a substitution, as you can see right here, is a substitution where we only have 2 reads, right? And they don't agree. They don't agree with the reference, but they don't also agree with each other. And finally, another example that we have here is an insertion, which we can say it's an insertion in the genome or a deletion in the reads that we have. But ultimately, this is a deletion that we need to identify as a kind of something that we trust, evidence that we trust, or something questionable. And so a few conclusions arise from this brief example. One of the conclusions is that the data can be more complex than it is presented. The other one is that some of the regions are going to be more trusted and others are going to be less trusted. And we need to understand how the data looks, but also what the analysis was that we used to be able to give this a biological interpretation. So because of these elements, we need to start looking deeper at the data and not only trusting whatever is being published, but really asking questions. And we ask questions of data using analytical methods. So let's take an example from this pandemic and briefly go through how to do that. So very early on, there was a paper that was published because everybody was concerned, where's this pandemic coming from? Was it engineered on purpose? What's really going on? And so a lot of people were asking these questions. And there was a paper that was called The Proximal Origins of SARS-CoV-2. And I'll share the link here in the chat, where there were several arguments presented about the potential reasoning of how people who specialize in research in bioinformatics for infectious diseases think about this data that emerged from this outbreak. And so this is what they told us. They told us that we have the nucleotide sequence, we have the amino acid sequence, we understand what proteins are being produced from this amino acid sequence, and therefore, if we can identify the relationship between multiple selected strains of this virus that we have fully sequenced, we can try to understand why this virus occurred as a natural outbreak and where could it potentially have come from? What was the potential intermediate and the original source of this pandemic? So how did they do this? The first example that I want to show you, is an example using the Tbone platform. And this will be a way for us to kind of explore the platform, but also continue talking about this subject. So the Tbone platform is something that you have access to through your account. And on this platform, we will look at an alignment. So how do we analyze and align these sequences to the reference genome, and what's called phylogenetic tree construction. So we're going to try and do some of these examples. But just so that you know, you will have access to this platform for the duration of this program, and you will have 5 terabytes of storage, where you will have a large number of capabilities, and you will have our technical support to help you get through some of these initial stages. So on the platform, there are 2 specific sections that we want to focus on first. One is alignment to a reference, right? So there we align reads to the reference genome, and we can identify specific variants, or alignment, multiple sequence alignment, where we have multiple sequences that we want to compare to each other, and that allows us to study relationships between them by looking at a phylogenetic tree. The first pipeline organizes these methods, right? So each button here represents a method. And as you can see, those methods will be constructed into a pipeline. So a series of methods organized sequentially, one after the other, given some input, make intermediate outputs, and then eventually produce a result, what we call a result. And we'll see that that could be the alignment file, or that could be a phylogenetic tree. So this pipeline will look like this. We'll take the FASTA files of the spike protein, and then we will load them into this pipeline. We will just use one button called multiple sequence alignment. Then we'll look at multiple sequence visualization and end. And as a result, we will have this interactive visualization that will allow us to navigate those sequences. Okay, so we will do this in the program. We will not spend too much time on this now to practically go through this, but then also after that, I will show you a pipeline that produces a phylogenetic tree. So let me briefly show you. I'm sure that most of you are already familiar with these methods, which is great, but just as a reminder, refresher, I will show you how this might work. Okay, so first of all, thank you, Ajastri, for sharing the link. If everyone can go to the server and let me know once you're in by putting in yes or 1 into the chat box. If you're having trouble logging in, right now we can figure out what the issue might be. So all of you should have the login information. If you don't, we'll be able to email that to you. So let's just go ahead and try that right now. Okay, so please let me know if you're having trouble logging in. Other than that, if you are in and you can see what I see, and you can go right here and see your email address. All right, great. Thank you, Katie. Katie is logged in. Anyone else? Okay. So Clement, maybe you can try later, okay, and let us know. Okay, Ekta is not able to. So maybe Ekta, could you please share the email address that you are trying to log in with? So Narika, are you here?
Yes, Ilya, I have taken Ekta's email address and I'll help her with that.
Perfect. All right, great. So once you're logged in, for those of you that were able to, what you want to do is you want to go right here into phylogenetic analysis. And I'll just show you briefly how this works. So you can either go into this section, and that will allow you to upload files and actually do the analysis, or you can click on this drop down and you will see some demo pipelines. And so in the demo pipelines, you will see right here SARS-CoV-2 origin, which is a pipeline that already has been finished. And you can simply click on each button, see what the button does, and then prepare a very simple pipeline. So what we'll do is start multiple sequence alignment, convert that multiple sequence alignment to amino acids. This is a way to look at specific positions in terms of mutation rate that helps us make more precise estimates of time based on mutation rate. Then we will combine all kinds of phylogeny methods together to prepare a phylogenetic tree. We will analyze all of these phylogenetic trees and select the average one between multiple methods, which is a method in bioinformatics that often is used when you have many different things to do the same step. And then we will run the pipeline. Okay. So again, start, multiple sequence alignment, multiple sequence alignment AA amino acid, select codon position MSA, all phylogeny method, tree space, end. Let me know when you have it. Okay, did anyone complete a pipeline like this? If you have, then you should go to click pipeline output files and find that you can download here a variety of files from this download folder. Okay, I'll just give it a minute. Later on, you'll be able to go back and we will actually show you where to find this analysis in greater detail and understand it. But this is just to introduce you to the tool today, right? So this is just a way to prepare a number of files that you could either visually see, like a PDF, download as a FASTA file, or potentially additional files like this full table TSV and the tree file. We'll talk about those in future sessions. Has anyone been able to run this demo pipeline, or is anyone struggling to find where exactly I found it? Okay, so again, let's go into areas of analysis. How do you log in? So I can log out real quick. You see, if I log out, you click on the login button right here. And you can use here your email address with whatever password you have to access the OmicsLogic portal. So if you have created your account, okay, you got to be logged in. So let me log in. So here I'm logged in. And now you click not on this, not on phylogenetic analysis, but you click down on this button and you go to SARS-CoV-2 origin. Okay, so Isha, were you able to click there? And
Yes, up till now I can, but when I come to phylogenetic tree, I wasn't able to click it. It wasn't showing.
so you don't see this demo?
So all this I can see, but when I come to the pipeline in the last step, I can't click that one. Yeah.
Oh, you can't click on this one?
This one. Yeah.
Okay, well, let's try that together. So here I click on start, multiple sequence alignment. multiple sequence alignment AA, select codon position, and then this should be highlighted like this. Okay, tree space, okay, end.
Okay, sir. Thank you.
All right. All right, excellent. Okay, very simple pipeline. Again, take a look at these output files. We're not going to go through them in detail. The objective of today's session is just to introduce you to some of these tools. But if you have completed what we just did, then you know that you can always go back and you will find under all of these different pipelines some examples that you can go through. And those examples will be used throughout the different sections that we will do, right? So take a look at them. Now, one of the pipelines that I made to kind of introduce some of the tools has an additional button for visualization, right? Because just having a FASTA file is not very useful. And I have this visualization, and in this visualization, I have a number of spike protein sequences aligned to each other, okay? So why are we looking at these sequences aligned to each other? The reason we are looking at them is because they can reveal a very specific pattern that the authors of this paper in Nature Medicine, kind of used to link this novel coronavirus in humans to a proximal origin, to where potentially could have come from. How did they do it? So first of all, they selected very specific sequences. They said, let's take human SARS-CoV-2, this new coronavirus, and let's take the bat rat G13, a genome that was sequenced very recently before this novel coronavirus emerged, a pangolin SARS-CoV-2 sequence, and then some previous sequences of SARS-CoV-1 from human and from bat. And so the first thing that they found is that the closest ones are human, bat, and pangolin, right? And here they noticed some very specific similarities that were present in some, but not all. And since the pangolin, bat, and human SARS-CoV-2 overall were more closely related to each other, then they said, well, in specific positions, pangolin actually resembles some important features for its ability to infect humans more than the rat G13 bat genome, right? And so that was their first conclusion. They said the receptor binding domain, we'll talk about that in a second, is much more closely resembled in the pangolin sequence to the human rather than the rat G13. And the second thing that they found was that even though that they found these important similarities in the receptor binding domain, in the polybasic cleavage site, which is another important section of that spike protein, they found this unique sequence that was only present in the human SARS-CoV-2, which is PRRAR sequence that was different from all the rest. Now, to get to these conclusions quickly, we need to master some tools. And these tools oftentimes include some of the simple things that we just did, like putting together buttons, or sometimes we need to go more in depth. And let's take a look at how you can practice those things as a part of this program as well.